Hello everyone, welcome to Physics Wallah English YouTube channel. I'm Shalini Somashekar, your botany teacher. So in today's session, we are going to be dealing with one of the most important chapters of your 12th grade syllabus called sexual reproduction in flowering plants. This is very important when it comes to your board exams as well as entrance exams like NEET. So in today's lecture, we are going to be covering all concepts of this chapter in just one shot. Alright, so brace yourselves guys, let's get today's session started. Since the chapter is called Sexual Reproduction in Flowering Plants, let's begin today's discussion by talking about flowers. Flowers come in so many different varieties, right? They come in so many different colors, shapes and sizes. And we humans over the course of time have come up with so many different ways to make use of them. We make use of flowers to worship our gods. Flowers are used to express love. It's also used during weddings. And when someone very close to us passes away, we make use of flowers to mourn their loss. And whenever you place a bunch of flowers on a table at home or at a corner in your house, it instantly adds a little extra beauty to that entire space, right? We sure have found so many different ways to make use of flowers. But why do plants produce them? Do plants produce flowers so that you and I can use it to decorate our hair or our homes? Obviously not. The reason why plants produce flowers is because in plants, flowers are the sexual reproductive organs. Now, what do we mean by sexual reproductive organs? Why do we say that human males' testes is the male reproductive organ and in human females, ovaries are the reproductive organs? It is because it is in these organs that the gametes are produced. Similarly, when you consider flowering plants, it is in the flowers that the male and female gametes get produced, because of which we call flowers the sexual reproductive organs. What is sexual reproduction basically about? Sexual reproduction involves the formation of gametes and the fusion of gametes. Whichever part or whichever organ in an organism's body is responsible for the production of those gametes, that organ is called the reproductive organ. In flowering plants, it is flowers that produce the gametes. Therefore, we call it the sexual reproductive organ. At this point, I want you to recollect some concepts that you've learned in your 11th grade syllabus in the Plant Kingdom chapter. In Plant Kingdom, we learnt about the various divisions: algae, bryophytes, pteridophytes, gymnosperms and angiosperms. Algae, bryophytes, pteridophytes, gymnosperms, they lack the ability to produce flowers. They do not produce flowers. It's only the angiosperms that have the ability to produce flowers, because of which they are also called flowering plants. And this chapter primarily focuses on how sexual reproduction takes place in flowering plants, which is none other than angiosperms. Alright, so in today's session, we're going to deep dive into how sexual reproduction takes place in flowering plants or angiosperms. So it is very important for us to understand the different parts of the reproductive organ flower before we can start to understand how reproduction takes place, right? So let's recollect the different parts of a flower that we've learned in morphology of flowering plants in 11th standard. So when you take a typical angiosperms, it has a couple of parts. The stalk of the flower is called the pedicel, right? At one end of the pedicel, it is attached to the plant. On the other hand, it will have, on the other end, it will have a broad base-like structure, which we call the thalamus. On this thalamus, several floral parts will be arranged in concentric circles, which we call whorls. Okay, it's called whorls. There are 4 whorls in a typical angiospermic plant. We will look at it from outside to inside. The outermost whorl or the outermost part of a flower is called the sepal. So these green colored leaf-like things that you're looking at here, these are the sepals. Collectively, the sepals are called calyx. So what is the function of this sepal in a flower? Whenever the flower has not yet fully bloomed, it is still in the bud condition, these sepals will protect whatever is lying inside it. Okay? Following the sepals, inside the sepals from the outside, the second layer or the second type of whorl is the petal. petal is usually the most attractive part of the flower. See, it is the bright, showy colored part of the flower, and these petals together constitute the corolla. Now, what is the function of petals? Petals are meant to attract the pollinators. Okay, so more about pollination and pollinators we'll learn going forward. For now, just remember it is meant to attract pollinators like bees and other insects. Now, what lies inside it is the male reproductive part. Each individual component here is called a stamen. What I'm highlighting here in yellow color, this is the stamen. And their entire whorl of stamens is what we refer to as the androecium. The stamen has two parts. The long slender stalk is called the filament, and the broad knob-like structure that you see on top is called the anther. Okay, now it is the stamen or the anther that is responsible for the formation of male gametes. So this is the male reproductive structure. Now let's talk about the innermost whorl, which is the pistil or gynoecium. Individual units of pistil or gynoecium are called carpels, and you can find three different parts there. You have the stigma, the long slender style, and the swollen ovary. Stigma, style, and ovary. Within the ovary, you have ovules, which will eventually become the seed. It is within the female reproductive part or the gynoecium that the female gametes are produced. Therefore, we can say that gynoecium is the female reproductive part of the flower. For sexual reproduction to take place, the presence and the production of male and female gametes is absolutely essential. Androecium's function is very, very important because this is what will produce the male gamete. Likewise, the presence of gynoecium is very important because this is what is going to produce the female gamete. Without gynoecium or androecium, sexual reproduction cannot take place. Therefore, the androecium and gynoecium, these two whorls are referred to as essential whorls. Essential whorls. Even in the absence of calyx and corolla, reproduction can take place. So they are called non-essential or accessory whorls. Okay. So in this chapter, we are going to be focusing on sexual reproduction. So we will focus more on androecium and gynoecium, because they are absolutely essential for reproduction. Even without calyx and corolla, reproduction can still take place, right? So, now let's moving on, focus on the structure of the stamen and the structure of pistil. So now let's take a look at one stamen. Each stamen has two distinct parts. There is a long slender stalk which we call the filament, and there is a knob-like structure on one end of the filament which we call the anther. The other end of the filament is attached to the thalamus. Now, I already mentioned that the stamen is the male reproductive part, or the androecium is the male reproductive part. That means this is the structure that will have to produce the male gametes. Among these two parts, it is the anther that produces the male gametes. Within the anther, the gametes are produced, or the gametophytes are produced. So, we call this the sterile part, the fertile part of the anther, and the filament is the sterile part of the anther. Now let's take a look at what are the different structures that you can see inside the anther. That is where the male gametes or the microspores or the pollen grains are produced. Okay, so this, when you take a section of this anther and look at it from above, here's what it's going to look like. Each anther will have two lobes. Okay, so here is the first lobe, this is one lobe and here you have the other lobe. Since the anther has two lobes, we say that it is bilobed. Bi means two, right? Within each lobe, you can find two cavities that are running vertically throughout the length of the anther. We call them pollen sacs. Okay, let me highlight it here. You can see 1, 2, 3, 4. So this runs throughout the length of the anther. Each lobe has two such pollen sacs, which we call theca. So we say that a typical angiosperm anther is dithecous. Since it has two lobes, there will be 4 such pollen sacs. So we say that the anther is tetrasporangiate. You must remember these terms. Okay. Now, these pollen sacs are also called microsporangia. What is sporangia? Sporangia are those structures that will have the ability to produce spores. Spore producing structures are called sporangia. Here we are calling the pollen sacs as microsporangia. That is because it is within these microsporangia that the pollen grains will be produced, right? So, since there are 4 such microsporangia, we say that the anther is tetrasporangiate. Tetra means 4. Okay. So, this is about the basic structure of an anther. Now, let's take a look at the transverse section of an anther. When you take this anther and take an extremely thin transverse or horizontal slice and look at it under the microscope, you can see that the wall of the anther has multiple layers. It is composed of 4 different types of cell layers. I'm going to tell you what each one of those are, starting from the outside. The outermost layer of cells that you see here is the epidermis. Remember, you've learnt about epidermis in anatomy of flowering plants in 11th grade. Any part of the plant, root, stem, leaves, the outermost layer is always going to be epidermis, and it is protective in function. So here you can see a single layered epidermis. Inside the epidermis, you can find another layer of cells, which I'm going to highlight or outline in orange color right now. That is called the endothecium. Endothecium also is a single layer. Inside the endothecium, you can find small thin-walled cells that may be 1 to 3 cell layers in thickness. These are called the middle layers. These can be 1 to 3 cell layers in thickness. Inside that, you have another layer of cells, which I will highlight in, let's say, green color. Okay. These cells are called tapetal cells, and that layer is called the tapetum. Tapetum usually contains dense cytoplasm, prominent nucleus, usually more than one nucleus also will be present. So over here, the function of the outer 3 layers, epidermis, endothecium and middle layers, is to offer protection to whatever is lying inside. Whereas tapetum is nutritive in function. That means it nourishes something. What does it nourish? It nourishes the developing pollen grains. I told you that the pollen grains will be formed within the anther. So pollen grains will form from a tissue called sporogenous tissue. So within the tapetum, you can find these cells here. It will form a homogeneous mass of cells. This is called the sporogenous tissue. Sporo relates to spore, microspore, genus means generating. So these sporogenous tissue, the cells in the sporogenous tissue will eventually become microspores or pollen grains. Okay. Now, these sporogenous tissue has cells that are called microspore mother cells, or in short you can call them MMCs. So microspore mother cells have an alternate name. They are also referred to as pollen mother cells. Pollen mother cells are also called PMCs. Okay, so this is the structure of the anther. In the wall there are 4 layers: epidermis, endothecium, middle layer, tapetum. In the wall, within that enclosing the it encloses the sporogenous tissue which is present at the center within the pollen sac. The cells of this sporogenous tissue are called microspore mother cells or pollen mother cells. This is basically the anatomy of a young anther. I told you that the outer 3 layers, epidermis, endothecium and middle layers, they offer protection to the anther. So they are basically protecting what is lying inside, very important tissue, sporogenous tissue. That is when the anther is young. When the anther matures, the sporogenous tissue would have formed microspores. At that time, the anther wall will break open on the sides and release the pollen grains that are produced inside by a process called dehiscence. So during the later stages, when the anther matures, the same 3 layers will help in dehiscence of the anther as well. They help in breaking the wall open so that the pollen grains that are produced inside can be dispersed easily. Alright, so that's about the anatomy of anther. Now let's talk about microsporogenesis. I told you that the sporogenous tissue is what will give rise to the microspores, right? So how exactly does that happen? Let's begin with the microspore mother cell. Each microspore mother cell or a pollen mother cell is a diploid cell. It consists of 2 sets of chromosomes, so we say that it is diploid. Now this diploid cell will undergo meiotic cell division, which is reductional division. When one pollen mother cell or microspore mother cell undergoes meiotic cell division, it will give rise to 4 cells. There's 1, 2, 3. Imagine there's one behind. So you will get 4 microspores. Each microspore is haploid. Since there are 4 microspores that are produced and that are kept together, held together, we call this a microspore tetrad. Eventually, these 4 spores that are held together and attached will dissociate, become individual cells and form the pollen grains. The pollen grains are haploid in nature. Within the pollen grains, the male gametes will form eventually. Okay, for now, when we talk about microsporogenesis, we are talking about how exactly microspores are generated or produced in the anther. So what basically happens here, the microspore mother cells that are present in the sporogenous tissue, they will undergo meiotic cell division and produce, excuse me, it's going to produce a microspore tetrad that has 4 haploid cells called microspores. Initially they are all present together, attached together next to each other. Eventually it will get separated to form pollen grains which are also haploid. So this process of formation of microspores from the microspore mother cells is what we call microsporogenesis. Now here you can see clearly how towards the end of maturation of the anther, the pollen grains are produced. You can see how the outer layers are no more intact. In between the 2 pollen sacs in each lobe, there is a slit and it has opened. Now the pollen grains are produced inside. They are going to be liberated, released out through these slits that are formed in between the pollen sacs. Okay, so here you're looking at a mature dehisced anther. Dehisced anther means it has opened up already to release the pollen grains. Okay, now let's talk about pollen grains. Pollen grains are usually microscopic. You do not see them and their structure with your naked eyes because they are extremely tiny. Talking about their size, they measure between 25 to 50 micrometers, microns in diameter. They're that tiny. You can see them under the microscope. They come in so many different colors and ornamentations. This image or illustration that I've added here, these show you different types of pollen grains. You can see how some of them are spiny, some of them have pores on their surface, right? So they come in many different varieties. Okay, so what exactly is a pollen grain when it comes to reproduction? Pollen grain is the male gametophyte. We've learnt about alternation of generation in plant kingdom. Whenever a spore germinates, it produces a gametophyte. Now the microspore is the male gametophyte because it is within the pollen grain that the male gametes will form. Since the pollen grain will carry the male gametes or will have male gametes within them, we say that it is the male gametophyte. Gametophyte is any structure that will produce the male gametes. In angiosperms, pollen grain will produce the male gametes. Therefore, it is the male gametophyte. Most students think that pollen grain itself is the gamete. Pollen grain is not the gamete. Within the pollen grain, you will have the male gamete. Alright? Now, talking about its shape. Like I already said, it comes in multiple varieties, but majority of them are spherical. So, they are generally spherical. And whenever you've seen hibiscus flowers, when you touch near the anther, a yellow powdery thing will drop on your fingers, right? So, that is nothing but the pollen grain. Now, let's talk about the structure of the pollen grain. A single pollen grain we'll consider now. One pollen grain in its wall will have two layers. The outer layer is thick and discontinuous. It is called the exine. It is called the exine. Let me draw it here. Okay, so this outer layer is called the exine. Exo means what? Outside. So exine is the outer layer. Exine in some places is incomplete. Okay, now that is why we say that it is discontinuous. This exine is made up of a substance called sporopollenin. What is sporopollenin? Many questions are frequently asked based on sporopollenin. It is a substance that is highly resistant to the treatment with very strong alkalis, strong acids. No enzymes can denature it, no hot temperature can denature it. So basically it is a highly resistant substance. Because of its high resistance, it can be retained even as fossils. Okay, so these are some points you have to remember. Sporopollenin, it is one of the most resistant organic materials that we know. It can withstand high temperature, strong acids and alkali. No enzyme so far we know that can degrade or break down sporopollenin, because of which it is preserved well as fossils. Okay, now I said that the outer layer or exine, which is made up of sporopollenin, is a discontinuous layer. There are some places in which sporopollenin is absent. So those regions are called the germ pores. That's about the exine. Now let's talk about the inner wall, which is the intine. Whatever I'm drawing in green color, this is the intine. The intine compared with the exine is a little thin, and also it is a continuous layer that is made up of cellulose and pectin. Similar to our other cells, like other plant cells, the cell wall here, the intine here is made up of cellulose and pectin. Now let's move on and talk about what lies inside the cytoplasm. Similar to all plant cells, within the cell wall you will have the cell membrane. Even over here you will have a cell membrane inside the cell wall. So you will have the cell membrane. This is the cell membrane. Within the cell membrane you will have the cytoplasm. When the pollen grain is mature, it will have two cells, one large cell and one smaller cell. The large cell is called the vegetative cell. It will be rich in reserve food, and there will be a smaller cell with dense cytoplasm, prominent nucleus that is spindle shaped. This is called the generative cell. Okay, so cytoplasm of pollen grain is surrounded by plasma membrane. It contains two cells when it is mature. One is the vegetative cell and the second one is the generative cell. Vegetative cell is bigger, it has abundant food reserve and a large irregularly shaped nucleus, like I already said. Now, the smaller generative cell, it floats in the cytoplasm of the vegetative cell. It is spindle shaped. It has a dense cytoplasm with a prominent nucleus, which I already drew and showed there. Now, let's talk about pollen grains and its uses. Some pollen grains have the ability to cause allergies in people. So when they are chronically exposed over the course of time, if they are constantly being exposed to these pollen grains in the environment that has the ability to cause them allergies, it can result in some chronic respiratory infections and illnesses such as asthma, bronchitis, etc. You may have heard about this Parthenium plant, right? So this Parthenium plant came to India when we were exporting wheat from other countries. So this came as a pollutant, and the pollen grain of Parthenium is known to cause allergies in a lot of people. So pollen grains uses that it is rich in nutrients. So it can be used as nutrient supplements. They are available in the form of tablets and syrups. And it is also claimed that by consuming pollen grains, race horses and athletes can increase their performance. Okay, so these are some uses of pollen grains. Now let's talk about pollen viability. The pollen grains carry the male gametes within them, right? So once the pollen grains are released from the anther, it is supposed to go and drop on the stigma and form a pollen tube inside the style and release the male gametes within them into the embryo sac of the female. For that, they will have to retain their viability. So once the pollen grain is released, it will not retain viability throughout its life. Once they are shed, they have to land on the stigma before they lose about their viability in order to bring about fertilization. Different species have different periods of viability. And what does it depend on? It also depends on temperature and humidity, and it also depends on the genetics. Like for example, there are some cereals such as rice and wheat where the viability is lost within the 30 minutes of release of the pollen grains. When you consider flowers belonging to some families like Leguminosae, Rosaceae and Solanaceae, there the pollen retain their viability for several months after release. Now, you may have heard of seed banks where the seeds are stored and you can go buy them. The seeds are preserved in a cold temperature. We've learnt about how sperms can be stored, how eggs can be stored, right? Similarly, pollen grain also can be stored by cryopreservation. So cryopreservation, what we basically do is we preserve pollen grains at -196 degrees Celsius in liquid nitrogen. So whenever there is a need, it can be used. Okay. So pollen banks are places where the pollen grains are stored in very low temperatures such as -196 degrees Celsius. Okay. Now, that's about pollen grains. Now, let's talk about gynoecium or the pistil, which is the female reproductive part of the flower. In a flower, you can have one carpel or more than one carpel. Carpel is the unit of gynoecium. Single units, individual units are called carpels. If there's only one carpel in a flower, we say that it is gynoecium is monocarpellary. If there is more than one carpel, we say that it is multicarpellary. When there is more than one carpel, it may be fused or it may be free. If the carpels are fused, we say that it is syncarpous. If the carpels are not fused, if they are free, we say it is apocarpous. There are these examples given to you in NCERT. Example for apocarpous is Michaelia, where the carpels, you can see there are so many, 1, 2, 3, 4, 5, 6, there are so many carpels that are not fused, they are free. Same if you consider for papaver, there are more than one carpels but they are fused. So it is syncarpous condition. Now let's talk about the different parts of the gynoecium. There are 3 distinct parts. You have the stigma, the style and the ovary. Stigma is the first part. It is the part where the pollen grains released from the anther will come and land. So stigma is the landing platform for pollen grains. Style is the long slender part that lies beneath the stigma. Ovary is the bottom bulged part or the swollen part of the pistil. Within the ovary, you can find ovules. So there is a cavity within the ovary which we call the ovarian locule. Within the ovarian locule, you can find a fleshy tissue which is called the placenta. Okay, so this is the placenta. You've learnt about placentation in 11th grade, right? So this is the placenta. Attached to this placenta, you will have one to many ovules. Ovules are what will become the seeds in the future. Okay, so these are the ovules. Ovules are one in plants such as wheat, paddy and mango. There are many ovules in papaya, watermelon and orchids. So if you don't know how many ovules are present in a flower, you can think of how many seeds are present in the fruit. In wheat, paddy, mango, you can find only one seed per fruit. Whereas in papaya, watermelon, there are many seeds in the fruit. So if there are more seeds in the same fruit, it indicates that it has more than one ovule, because eventually after fertilization, ovule will become the seed, ovary will become the fruit. Okay. So now let's talk about the ovule, one individual ovule. Okay, so let's just focus on one ovule here. The ovule is called the megasporangium. Why do we call it the megasporangium? It is the sporangium because it is within the ovule that the megaspore will be produced. Now, ovule will have a stalk and the stalk is called the funicle. Stalk of the ovule is called the funicle. Ovules come in so many different varieties. Okay, so if this is the stalk, ovule will be like this. But here what you're looking at is an inverted ovule. In angiosperms, mostly we find inverted ovules that are called anatropous ovules. Okay, so this is an illustration of an anatropous ovule. So over here, you can see that there is a point at which the body of the ovule is fusing with the stalk. Okay, so here the body of the ovule is fusing with this stalk. So this point or this portion is called the hilum. Ovules are covered by two protective coverings which we call the integuments. See, here you have one integument. This is the first covering. Inside that, you have the second integument. The integument towards the outside is called the outer integument, and the integument that is present towards the inside is called the inner integument. These integuments do not fully cover the ovule. There is a region where it is uncovered or a small pore is present. That pore is called the micropyle. Okay. Now, within the ovule, you can find a homogeneous mass of cells which is called the nucellus, not nucleus, nucellus. Within the nucellus will lie what is called the embryo sac or the female gametophyte. Within this female gametophyte, the female gamete will be present. Now, the part or the half of the ovule that is present towards the micropyle is called the micropylar pole, and the opposite end is called the chalazal pole. These are the different parts of the ovule. Now, let's learn about how megasporogenesis will take place. From the nucellus, the megaspore will be produced. What happens during megasporogenesis is, towards the micropylar end, there is one cell that will differentiate from the other nucellar cells, and it will become larger in size, as you can see here. The surrounding cells here, these are the nucellar cells. Among them was another nucellar cell that has kind of changed its structure compared to the other cells. It's become differentiated. It has denser cytoplasm and a prominent nucleus. Now, this cell is called the megaspore mother cell. There it was microspore mother cell. Over here we call it the megaspore mother cell. This megaspore mother cell, similar to microspore mother cell, will undergo meiotic cell division. After the first meiotic division is undergone, two haploid nuclei are formed. This is called a megaspore dyad. After meiosis II, each of these cells will undergo meiosis II and form 4 cells that are usually linearly arranged one on top of another, and this is called the megaspore tetrad. The difference between megasporogenesis and microsporogenesis is, in microsporogenesis, all the sporogenous cells or the microspore mother cells will undergo meiotic cell division to produce pollen grains. But over here, generally in one ovule, only one of the nucellar cells will undergo some differentiation to form megaspore mother cell. Only that will undergo meiotic cell division to produce 4 megaspores. Okay, so this is called the megaspore tetrad. Now, among these 4 megaspores, 3 megaspores that are present towards the chalazal end will usually degenerate. And there will be only one functional megaspore. And this one functional megaspore will form the female gametophyte or embryo sac. Okay, since over here of the 4 megaspores that are formed, 3 will degenerate, only 1 will remain functional, that will form the embryo sac. We call the development of embryo sac as monosporic development. So this is monosporic development of embryo sac. Okay, now let's learn about how exactly the embryo sac or the female gametophyte gets formed. Now we had one cell, haploid cell. This was the megaspore. Now this haploid megaspore will undergo mitotic division to form 2 nuclei. Initially only karyokinesis will happen, nuclear division will happen. Eventually, cell plate formation or cytokinesis will take place. So from one nucleus, two nuclei divide. Each of these nuclei further undergoes another mitotic division to form two nuclei at each pole. Further, those two nuclei will undergo another set of mitotic division so that at each pole you have four nuclei. Remember, all of these nuclei are haploid. This is mitotic cell division where the ploidy will be retained. So, of the four cells that are present towards each pole, one nucleus will migrate towards the center. After that, cell wall formation will take place. So, at the micropylar end, towards the micropyle end, you will find three cells. Towards the chalazal end, you will find three cells. At the center, you will find one cell. Towards the micropylar end and chalazal end, you will have three cells, each with one haploid nucleus. In the central cell, you will have two haploid nuclei that have migrated from the opposite poles. We call these two nuclei as polar nuclei. So when you look at the final arrangement of cells in an embryo sac, you can see that the chalazal end will have three cells, 1, 2, 3. Each cell contains one haploid nucleus, and these cells are called antipodals. On the opposite end, you will have three cells that are called egg apparatus. In this egg apparatus, you will have three cells. Two cells that are lining towards the sides, they are called the synergids. In between the synergids in the center, you will have the most important part, which is the egg cell or the female gamete. All of these are haploid. Towards the base of these synergids, you can find cytoplasmic finger-like projections like this, that is called filiform apparatus. Eventually, the pollen tube that carries male gamete should come and discharge its contents inside. So, this filiform apparatus will kind of guide the pollen tube into the embryo sac. Now, let's move to the central cell. The central cell here is the largest cell in the embryo sac, and within the central cell, you will find two haploid nuclei that are called the polar nuclei. So, the mature embryo sac in angiosperms will have 6 cells towards poles, 3 each, one large central cell. Totally, there are 7 cells and 8 nuclei. Why are there 8 nuclei in 7 cells? Because one cell, that is a central cell, will contain 2 nuclei. Okay, so the embryo sac is 7-celled and 8 nucleate in angiosperms. Now, let's talk about pollination. Pollination is the transfer of pollen grains from the anther to the stigma. This transfer may happen within the same flower, in between two flowers, or in between two plants altogether. Okay, depending upon how pollen transfer takes place, we can classify or categorize different types of pollination. Firstly, let's learn about autogamy. Auto means self, gamy means marriage. So in autogamy, as the name itself indicates, the same flower, if you take this as a plant, let's say this is the flower. In this flower, you have the anther and you have the pistil. The pollen grains from the same flower will deposit on the stigma of the same flower. Such a type of pollination is called autogamy. Now, this can happen either in Casmogamous flowers or in Casmogamous flowers are the normal flowers that are open. After a point, the bud will bloom and the flower will open. In some plants such as Viola and Camelina, you find two different types of flowers. Flowers that are open and flowers that are closed that never open. Usually these are found below the soil. They don't open at all. Such flowers are called cleistogamous flowers. What happens in cleistogamous flowers is, if the flower does not open at all, then the pollen from the same plant, same flower, will fall on the stigma of the same flower and cause self-pollination. One advantage of cleistogamous flowers is, over here you don't need a pollinator because the floral parts are placed very close to each other. Even in the absence of pollinator, seed set will be assured. Now let's learn about the second type of pollination, which is called geitonogamy. Over here, if you take a plant, let's say this is a flower, this is a flower. Over here, the transfer of pollen grains happens between two flowers belonging to the same plant. The pollen from one flower drops on the stigma of another flower belonging to the same plant. Such type of pollination is called geitonogamy. Now let's talk about the third type, which is called xenogamy. Xenogamy is nothing but cross-pollination that happens between two different plants. In xenogamy, you have two plants . This is the flower, let's say. The pollen from one flower drops to the stigma of another flower in a different plant. Such a type of pollination is called xenogamy. This is actually cross-pollination. Okay. Now, pollen grains cannot themselves walk or move around because they do not have locomotory structures. So, pollen grains will have to be transported from the anther to the stigma. This can be done by means of biotic agents or abiotic agents. Biotic agents are living agents, living creatures that are helping in the process of pollination or the transfer of pollen grains. Abiotic agents are agents like wind and water, which will carry the pollen grains along with them in their wind current or water current. Okay, so let's talk about individually each of these different types of pollinating agents. If it is happening by wind, we call it anemophily. If it happens by water, we call it hydrophily. If it happens because of biotic agents like animals, we call it zoophily. Okay. Now let's talk about wind pollinated flowers. The plants that in which we see wind pollination, you can see that they have specific adaptive measures or they have specific strategies evolved in order to make sure that the pollination happens efficiently. The pollen grains in plants that are wind pollinated are produced in very large numbers. Even if the number of ovules per ovary is only 1, they will produce pollen grains in huge numbers, sometimes in thousands. And these pollen grains are very light in weight and they are not sticky. Now, why should it be light in weight and not sticky? Because these are being transported by wind. If they are very heavy, then because of its density, it will fall off. It cannot be transported by wind. If it is sticky, it will get stuck onto random surfaces on the way and may not reach the destination. Therefore, the pollen grains in such wind pollinated plants or flowers are very light in weight and non-sticky. How about the stamens? The stamens are very well exposed. Because the stamens will be very exposed, it becomes easy for the pollen grains to be dispersed. How are the stigmas? Stigma is very large and it is feathery in nature. This you have to remember. Why should it be feathery? Because pollen grain is being transported by wind. In order to trap that airborne pollen grains. If it is feathery, it can trap it very easily. Ovules, usually in plants that are wind pollinated like grasses, rose and rice and all of that, there is only one ovule per ovary. Okay, so that is the modification in ovules. Usually flowers will be very small in size and they will be packed in the form of an inflorescence. You can see this in wheat and rice plants. For example, when you consider the cob of corn, the sweet corn that you eat, you know, towards the end you have these hair-like structures, right? So they are basically the stigma and style of the plant. The tassels that you see are nothing but the stigma and style, and they are present in order to trap the pollen grains that are coming in the wind. This was a question asked in NEET in 2023. So this kind of pollination is common in grasses, like I already mentioned. Talking about water pollination. Water pollination is very, very rare in nature. You don't find it very frequently. Even in plants that are living in water, aquatic plants, pollination by water is very, very rare. If you consider all of the angiosperms, mostly water pollination is limited to monocots, and that too only in 30 genera, about 30 genera. But if you consider lower plants like bryophytes and pteridophytes, you know that water is very, very important for the male gametes to be transported, because of which their distribution is also restricted only to areas that are moist, right? So if we talk about angiosperms, not all plants that are aquatic use water for pollination. If their flowers are produced towards the surface, above the surface, like in lotus and water lily, wind or insects only will pollinate it. Some examples of angiosperms where water pollination takes place is Vallisneria and Hydrilla. Both of these are aquatic plants that are found in fresh waters. And then we have Zostera, which is a marine angiosperm. It is also called seagrass. Okay, so in these plants we can see water pollination. Now let's understand how water pollination happens in Vallisneria. Here is an illustration. You can see how the plants are submerged, they are present, they are rooted inside the water body, right? They formed roots and they have settled inside the water body. Over here you can find two different types of plants. There's a male plant and there's a female plant. The male plant produces the male flowers. These male flowers or the pollen grains will float on the surface of water. The female plant will produce the female flowers, which will have kind of coiled pedicel. So once the female flower is mature, it will uncoil and reach the surface of water. In the surface of water, the male flowers are floating. They will release the pollen grains and pollination will take place. After pollination has taken place, these female flowers will coil back and come inside the water. Okay, so this is how pollination takes place in Vallisneria. In seagrasses, what happens is, even in seagrasses, the plants are submerged. They are present inside the water. The pollen grains that are produced are long and ribbon-like, and they do not come to the surface of water. They are just as dense as water, so they will float within the water itself, and they will find the stigma and pollinate the stigma. Usually what happens is, the pollen grains that are produced in plants that perform wind pollination, usually because pollination will happen by water, outside the pollen grains there is a layer of mucilage that will prevent the pollen grain from wetting. Okay? Now let's talk about zoophily or pollination by animals. There are so many different types of animals that will help in pollination. Some examples include bees, butterflies, flies, beetles, wasps, ants, moths. You can see how so many of them are insects. Sometimes birds, bats and bigger animals like lemurs and tree-dwelling rodents, sometimes lizards also help in pollination. If an animal is helping in pollination, we call it zoophily. Specifically, insects will help in pollination, then we call it entomophily. Usually, among insects, bees are the most important pollinators for several species of plants. Okay, so usually these insect pollinated flowers show some characteristic features. They are very large, colorful, fragrant and rich in nectar. If a flower is expecting an animal to come and pollinate it, help it in its continuation of generation, then the flower will have to be attractive enough for the pollinators to come and visit, right? Therefore, the flowers in plants that are pollinated by insects, you can see that the flowers will be very large, showy and colorful and bright. Sometimes if the flower size is small, they will arrange compactly to form an inflorescence. Whenever the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous, to make them attractive for the pollinators. Animals are attracted to flowers by color and by fragrance. That is why they are brightly colored. Fragrance, it doesn't necessarily have to be like a pleasant smell. Sometimes bad smell also is produced by flowers. It will attract the dung flies and flies that will rot, that will grow on rotting meat. So it will smell very weirdly, and it will emit a foul odor to attract such insects. Okay, so in order to make sure that the pollinators, animals insect the flowers and bring about pollination, these flowers will have to give rewards to the pollinators. What are the rewards that the flowers will give to them? It is nectar and pollen grains. So to sustain the animal visits, the flowers will produce nectar and pollen grains that are the floral rewards. What happens is, insects, in order to suck the nectar from the flower or in order to take the pollen grains from the flower, it will visit the flower. Once it visits the flower, the pollen grains that are produced in the flower, that will be dusted on the back of the insect or on the body surface of the insect. Usually the pollen grains that are produced by insect pollinated flowers are sticky in nature. So it will stick to the body of the insect. Now after this insect has taken the nectar or the pollen grain or both from this flower, it will move to another flower to do the same thing. When it does that, the pollen grains that have been dropped on the surface, body surface of this insect, will be transported to the stigma of another flower. In that way, it will bring about pollination. Usually, some flowers even produce a safe place for the insects to lay their eggs. There is a plant called Amorphophallus. Amorphophallus is the flower. It produces a very tall flower which is about 6 meters in height, much taller than me. So, I'm sorry, 6 feet in height, not 6 meters, which is much taller than most of us. So, in that flower will allow some insects to lay eggs in them. Okay, so when the insects will come to these flowers to lay their eggs, it will bring about pollination. There's another wonderful example that we see in nature where a symbiotic relationship exists between a moth and a flower. The moth is called the yucca moth, and the flower and the plant is called yucca, and the moth that is symbiotically associated with it is also called the yucca moth. So what happens here is, these two species are dependent on each other for reproduction. If the insect yucca moth has to lay eggs, it will go and lay eggs inside the flower, and only this moth can bring about pollination in that particular type of flower. So what happens, it will go and lay eggs inside the ovaries of that flower and it will come outside. Once fertilization, after this has pollinated that flower, fertilization will take place and then the egg will start developing. So once after fertilization, the ovule will start developing into the seed. As the ovule develops into the seed, the eggs that are laid inside the flower, the moth's eggs that have been laid inside the flower, they will also break open and the larva will come out. In this way, they are both helping each other out in reproduction. Okay, so these are some examples. Now we learnt about self-pollination and cross-pollination. Self-pollination, if it happens over a period of time continuously, then it will result in something called inbreeding depression. Continued self-pollination will cause inbreeding depression. Now what do we mean by inbreeding depression? You may have seen how even in humans, you are advised not to get married to your very close relatives or first cousins. You may have noticed how in some families, when their first cousins get married to each other, the children that are born are either mentally retarded or they are deaf and mute, they can't speak, they can't hear. These kind of abnormalities we see when there is inbreeding. Inbreeding, you can think of it as very close relatives breeding. So this is bad for any plant. If it cannot survive properly, if it is not hardy enough, it is bad for the plant. In order to prevent that, plants have come up with some genius mechanisms. We say that plants don't have brains and they are not very smart, but despite lacking brains, they've come up with some genius mechanisms that can prevent this self-pollination and promote cross-pollination. So that is what we call as outbreeding device. They prevent inbreeding and promote outbreeding. So devices that discourage self-pollination and encourage cross-pollination are referred to as outbreeding devices. What are some of those mechanisms we'll see here? In some plants, the time at which the pollen is released, the stigma of the same plant will not mature by then. In order for pollen to form a pollen tube on this after deposition on the stigma, the pollen and the stigma have to mature simultaneously. If they are not maturing simultaneously, then the stigma will not allow the formation of pollen tube. That is one. Secondly, the anther and stigma are placed at different positions. What if the anther is shorter and the stigma is much taller or longer than the anther? Even if the anther matures, there are fewer chances of the anther going and falling on the stigma of the same flower. That also prevents autogamy. Now, self-incompatibility. There is a genetic mechanism that will prevent self-pollination from the same flower or from the same plant. What basically happens here is, if on the stigma a pollen grain from the same flower or the same plant is dropped or deposited, it will prevent the germination of the pollen or the formation of the pollen tube. Okay, so the pollen tube will not form at all, or it will not grow deep inside the style. So how does this happen? Whenever a pollen tube has to form after deposition on the stigma communicates with the pollen grain. Okay, the stigma by means of chemicals will talk to the chemicals present in the pollen grain. Only when there is compatibility, the stigma will allow the pollen grain to germinate and form the pollen tube. Otherwise, you can imagine how in nature there are so many different species of plants. All of them are producing the pollen grains, releasing them out in the environment. It is not like on the stigma of hibiscus plant, only pollen grain from hibiscus plant will come and fall, right? There are so many other plants that are releasing the pollen grains in the air. Any pollen grain can come and fall on it. But it knows which pollen grain it has to allow to germinate and form a pollen tube. So for that, communication happens between them in the form of chemicals. So in such a case, in one of the mechanisms of inbreeding, outbreeding devices, the pollen grain, if it is from the same plant or from the same flower, these stigma will not accept it. It will not allow it to germinate and form a pollen tube. Okay, so this is the third point. Fourth point is to make unisexual flowers, like separate male flower and a separate female flower. Now both the male flower and female flower may be present on the same plant, which we call monoecious condition, or they may be present in two different plants. Like there's a separate male plant producing male flower, separate female plant producing female flower. We call it dioecious. Monoecious can be seen in castor and maize, where the plant is same, but in the same plant male flowers, female flowers are different. In papaya, you can see how there's a separate male plant and a separate female plant. Male plant produces male flower, female plant produces female flower. If it is monoecious, you can prevent autogamy, pollination within the same flower, but you cannot prevent geitonogamy, because within the same flower you have both male and female. Within the same plant you have both male and female flowers. We need to talk about dioecious conditions. Since the plants themselves are different, here autogamy as well as geitonogamy both can be prevented. Okay. Now let's talk about pollen pistil interaction. I already said that whenever a pollen grain falls on the stigma and pollen grain communicate by means of chemicals, and whenever there is compatibility, the pollen grain will allow the germination of the stigma will allow the germination of the pollen grain and the pollen tube will form. Okay, so we saw how in a pollen grain there was an outer exine and an inner entine, right? So through the germ pores that are present in the pollen grain, the pollen tube will start forming. So it forms, it comes through the style and enters the embryo sac, usually from the micropylar end. From the embryo sac, once it is inside the embryo sac, it will release the male gametes inside the embryo sac. Okay. Now let's talk about artificial hybridization. You know how in agriculture you want specific combinations of characters, right? For example, let's say there is a plant that produces like some beautiful colored flowers. Okay, so this plant produces flowers that are very beautiful, pretty to look at. It's large in size, nice colored flowers. over here the flower quality is great, but there is no disease resistance to this plant. So this plant or this variety of plant is prone to diseases. So it is not disease resistant. There is another plant of the same species which will produce flowers that are not so great to look at. It will not fetch a lot of money. They are very small in size, though of the same plant, but this is extremely resistant to diseases. Now, a farmer has to bring this and this property together in one plant. They can do it by artificial hybridization. What they do in artificial hybridization is they consider one of the two flowers as the female flower. Okay, let's say both of these are bisexual. They have the male as well as the female part. They will consider one of these two plants as the female. They will designate one plant as the female. In this female plant, if you're considering this as the female plant, you have to remove the male part of this plant. Before the flower matures and blooms, they will remove the anther from this flower. Anther is removed. Removal of anther is called emasculation. Masculine means maleness. Emasculation is removal of male structures. So over here, this flower, which is designated as female, from this the anther is removed by a process called emasculation. After the anther is removed, this flower is covered with a polythene bag, so that unwanted pollen will not contaminate this flower. So the second step is bagging. Next, you take pollen grains from this flower, anther from this flower, and dust the pollen grains onto this flower. Remove the bag, dust the pollen grains, desired pollen grain on top of this flower. That is called artificial hybridization, or pollination is performed artificially. After that, you cover it with the bag again. That is called rebagging, to prevent contamination of unwanted pollen. So when you do this, the pollen grains from the required variety of plant will be deposited on the stigma of the plant that has been designated as female. Okay, so this process is called artificial hybridization. Whenever this kind of cross is done, you can't for sure expect that the flower that is bright and is of economical importance will this character will combine with this character. It is not guaranteed. It might be the other way around also. So this takes a lot of trial and error and will consume a lot of time, energy and labor. Okay, so this is basically what artificial hybridization is. Now let's talk about double fertilization. Now I said when we spoke about pollen grains that the pollen grains have two cells in them. There is a vegetative cell and a generative cell. Vegetative cell is the large cell, generative cell is the smaller cell. When the pollen grain is released, it is released in the two-cell stage, usually released in the two-cell stage. What happens is, sometimes it is released in the three-cell stage. What happens in a three-cell stage is, you have the pollen grain, you have the vegetative cell, you have the generative cell. This generative cell will undergo cell division to give rise to two male gametes that are haploid. So within the pollen grain, you have a vegetative cell, and within the vegetative cell, you will have two male gametes. That is why the pollen grain is called the male gametophyte. Okay, so if the pollen grain is released in the two-cell stage, after it deposits on the stigma, it will divide and form two male gametes. Sometimes in some species, pollen grain is released in the three-cell stage itself. Even before the release, this division could have taken place. So within the pollen grain, you have two male gametes now. Now these two male gametes will fuse with two different cells in the female gamete and female gametophyte or embryo sac, and that is called double fertilization. So what basically happens here is, let's draw the female embryo sac. If you take this as the chalazal end and this as the micropylar pole, towards the chalazal end you have three cells that are called the antipodals. Towards the micropylar end you have two synergids with their filiform apparatus, and you have a central egg cell. In the center you have a central cell with two polar nuclei. Okay, now when pollination takes place, the pollen grain will come and settle on the stigma. If it is compatible, it will form a pollen tube. Through this pollen tube, the male gametes, the two male gametes will be getting transported into the embryo sac. So the pollen tube is guided by the synergids. So if this is the pollen tube, this pollen tube will come inside and it will release the two male gametes. One of the male gametes fuses with the egg cell, the other male gamete fuses with the two polar nuclei. So fusion is happening twice, that is why it's called double fertilization. What happens is, in the first fertilization or fusion, we call it syngamy. In syngamy, one male gamete, that is haploid, fuses with one female gamete which is also haploid to form a zygote that is diploid. This zygote will eventually divide to form the embryo. This is called syngamy. In the second fertilization, which is called triple fusion, three haploid nuclei fuse. You have the second male gamete, this second male gamete, which is haploid, will fuse with the two polar nuclei which are both haploid and form a triploid structure which is called primary endosperm nucleus. In short, it is called PEN. This is triploid. Eventually this will divide and form the endosperm. So the endosperm is also triploid. Here the embryo is diploid. So these two fertilization reactions happen in angiosperms, and it's the characteristic feature of angiosperms. It is called double fertilization. Now let's talk about endosperm and embryo specifically. Endosperm is the triploid nutritive tissue. Once within the seed, within the ovule, endosperm and the double fertilization happens, endosperm and embryo development will start. Usually before embryo develops, endosperm development will be fast, because for the developing embryo, nutritive tissue is required. Endosperm will provide nutrition for the developing embryo. The embryo is what will become the future plant. You know, when you sow the seed, a new plant will arise. The new plant will arise because of the embryo. And in order for the embryo to develop, food is required. Within a seed, you do not have leaves to photosynthesize, right? So the nutritive tissue that will help in the development of the embryo is the endosperm. So endosperm is triploid in nature. It is happening as a result of triple fusion. Usually what happens is endosperm, once the primary endosperm nucleus is formed, the primary endosperm cell will start undergoing mitotic divisions. Usually it will be free nuclear divisions where karyokinesis is taking place. Immediately after karyokinesis, cytokinesis will not take place. As a result, it will form a free nuclear endosperm, which is multinucleate. The tender coconut water that we drink, it is an example for free nuclear endosperm. Eventually, cross walls will be formed between these nuclei and the endosperm will become cellular. The coconut kernel that we see, that is an example for cellular endosperm. Now, depending upon when the seed is mature, if endosperm still persists or if all of it is used up, we can classify seeds into two types, which is endospermic and non-endospermic. Or albuminous also we say. In this, even in mature seed, endosperm will be present. If it is non-endospermic, or X-albuminous, endosperm will be absent at maturity. Usually, endospermic seeds you can see in monocots. In non-endospermic seeds, you can see in dicots. But there are dicots that are endospermic, there are monocots that are non-endospermic. For example, castor is a dicot plant that has endosperm. Orchids are monocot plants whose seeds lack endosperm. So these are some examples. That's about the endosperm. Now let's talk about the embryo. Embryogeny is the process or the development of embryo. Over here you have primary endosperm nucleus, pen that I said, this will develop into the endosperm, and here the zygote will develop into the embryo. Once double fertilization takes place, the other cells in the embryo sac will start degenerating. You can see how the synergids and the antipodals have degenerated. So it is this zygote which will give rise to the embryo. Okay, so this zygote will start undergoing cell divisions and increase the number of cells. The development of embryo is similar. I mean, it has some differences in monocots and dicots, but the initial few steps are very similar. So after the zygote starts dividing, initially it will form a globular embryo which is spherical in nature. Later it will become heart-shaped. Eventually it will mature to become an embryo like this. Now let's take a look at the embryo. This is a dicot embryo. You can see how there are two cotyledons. Okay, so for example, in dicot embryo you will have two cotyledons. It is shown here. This is one cotyledon, this is one cotyledon. There will be an axis to which the cotyledons are attached. The portion of the axis above this point of attachment is called the epicotyle, and the portion of the axis below this point of attachment is called the hypocotyle. The tip of epicotyle is called the plumule. This plumule will eventually give rise to the shoot system. The tip of the hypocotyle is called the radicle, and this radicle will eventually give rise to the root system. Okay, so here you can see this is the axis, this is the point of attachment, this is the plumule. Here this is the radicle, hypocotyle, this is the radicle. Radicle is covered by a root cap. That is the story of dicot embryo. In monocot embryo you have a single cotyledon. Monocot itself means single cotyledon, right? So this single cotyledon is shield shaped and large, and it is called the scutellum. Okay, so this large single cotyledon is called the scutellum. So this is the embryonal axis here. This is the point of attachment of the cotyledon. Above this, whatever is there, this is the epicotyle. Below this, what is there? This is the hypocotyle. Tip of the hypocotyle, you have the radicle. Tip of the epicotyle, you have the plumule. Over here, the plumule and coleoptile are covered by layers, protective layers that are not differentiated. The layer that covers the plumule is called the coleoptile. The layer that covers the radicle is called the coleorhiza. Okay, see, it's given here, coleorhiza. So these are the different parts of monocot embryo. Now let's talk about seed. Seed is the final product of sexual reproduction. Both double fertilization events happened within the ovule, within the embryo sac, right? Embryo sac was present within the ovule. So ovule will develop into the seed. So seed is the final product of sexual reproduction. A seed can be described as a fertilized ovule as well. Where are seeds formed? They are formed inside of fruits. Typically, what are the different parts of the seed? Seed will contain two seed coats. Which are the seed coats? In the ovule, whatever integuments were present, they will become dry and they will become the seed coats. You will have cotyledons. If it's dicot, there will be two cotyledons. If it's monocot, you will have one cotyledon. And you will have an embryo axis to which the cotyledons are attached. You have the radicle, plumule and all of that. The cotyledons are simple structures. Usually they are thick and swollen and they store reserve food. In mature seeds, albumin may be present or endosperm may be present or absent. I gave you examples of this already. One thing we must remember here is about perisperm. I told you that the nucellus is present inside the ovule. One of the cells of the nucellus will differentiate, become megaspore mother cell, produce megaspores, 3 will degenerate, 1 will remain functional. That functional megaspore will become the embryo sac. While all of this is happening, while double fertilization is happening, endosperm is developing, embryo is developing, nucellus will start to degenerate or it will get used up in all of this. Sometimes in some plants such as pepper and beet, what happens is black pepper and beet, even within the seeds you can see a thin layer of nucellus still persisting. That remnant persistent nucleus is known as perisperm. Peri means around, sperma means seed. So outer one layer you can see of nucellus being still present. That is called the perisperm. You can observe that in these plants. Okay. Yeah, like I already mentioned, the integuments will become the seed coats. The small pore that was present in between the integuments, it did not fully complete. There was a pore present in the ovule. Same pore will be present in the seed also. It is very important because if the seed has to respire, it needs oxygen, it needs water. So all of that it can get through this pore. Otherwise, if it's fully closed, that cannot happen. If a seed has to germinate, it requires water. So water can enter the seed, the living part of the seed, through the micropyle. As the seed will mature, it will kind of become dehydrated. It will start losing a lot of water, which is what will help you store the seeds for a long time. So generally the metabolic rate of the embryo also will slow down. So in some seeds, the embryo will enter a state of inactivity called dormancy. Whenever the conditions are not favorable for its germination, it will become inactive, which we call dormant. On the return of favorable conditions where the temperature is fine, the moisture is fine, there's sufficient oxygen, carbon dioxide, when all of that is favorable for its germination, then the seed will germinate. Okay. Now, there are several advantages of seeds. What are the advantages? You know how many fruits have evolved mechanisms for seed dispersal, right? The plant may be produced here, the seed may be dispersed elsewhere, and because of that, this particular plant will be that type of plant will be formed elsewhere. For example, if there's a, let's say, a bird that feeds on a fruit, it has digested the seed, it goes and defecates somewhere else, there that seed will germinate and become a new plant. Like that, for dispersal of seeds, there are various mechanisms. There are some plants that will just, where the fruits will just burst open and release all of the seeds outside so that they find suitable substratum for germination. So they have evolved mechanisms for dispersal of seeds to distant new habitats. Within the seed, there is sufficient food reserve. So until the leaves are produced, the germination happens, new leaves are formed, until they can perform photosynthesis, there's enough nutritive reserve within the seed. The seed coat, there are two seed coats that are present outside the seed, which offers protection to the embryo that is lying inside. Because it's a product of sexual reproduction, definitely there will be newer genetic recombinations that have formed. It is the basis of agriculture. You cannot even think about agriculture without seeds, right? So, talking about dehydration and dormancy, Dehydration and dormancy is also very important. Otherwise, if the seeds are germinating as soon as they are formed, you can't store them, right? Seeds are, we store seeds at home also, right? That is because they are dormant and they remain inactive for a specific period of time, and because of that we can have food throughout the year. So seed viability, talking about seed viability, in few species seeds lose viability within a few months. When we spoke about pollen grains, I told you about pollen viability. After the pollen grain has released, it will retain the ability to form a pollen tube on finding a suitable stigma only for a particular period. That is pollen viability. Similarly, even we have seed viability, where the seed will retain the ability to germinate only for a specific period. In some species, they lose their viability within a few months. There are large number of species of plants where the seed viability is present for several years. So, some seeds can remain alive for hundreds, not only hundreds, thousands of years. There are many records that are available for this. 2 examples are given to you in your syllabus. The oldest seed that we know was of Lupinus articus. This is the name of a plant. It was excavated from the Arctic tundra, which is a very cold region. So, this particular seed was germinated after an estimated 10,000 years of dormancy. For that long it was present within the ice, it remained dormant. On the return of the favorable conditions, we saw that it germinated after 10,000 years of dormancy. Okay. Another thing is a 2,000-year-old viable seed of a plant called date palm, scientific name Phoenix dactylifera, was discovered during an archaeological excavation that took place in the King Herod's palace, which is near Dead Sea. These are specific examples that you will have to remember. Now let's talk about the fruit. Ovule became the seed. As the ovule is becoming the seed, the ovary will transform into the fruit. The wall of the ovary will become the wall of the fruit, and we call the wall of the fruit as the pericarp. Some fruits are fleshy, some fruits are dry. Examples for fleshy fruits include guava, orange, mango. Dry fruits are groundnut, mustard. Example, these are fruits in which the pericarp is not fleshy, so we call them dry fruits. In most plants, by the time the seed will develop, the fruit also would have started to form, and other parts of the plant will have started to degenerate and fall off. Usually fruit is formed from the ovary. The part that we consume, edible part, is formed from the ovary. If it is formed from the ovary, we refer to it as true fruits. There are some examples like strawberry and apple, where ovary is not the main edible part of the fruit, but some other part of the plant which is called the thalamus. So you saw that the thalamus was the lower base on which the other floral whorls were arranged, right? In plants like apple and strawberry, cashew, you can see how the thalamus is forming the edible part of the plant. Edible part of the fruit. So such fruits are called false fruits. Remember these examples. Now again, in most species, after fertilization, seeds will form from the ovule, fruit will form from the ovary wall. But in some plants, even without fertilization, fruits can form, and such fruits will be seedless. They will not have any seeds. Such fruits are called parthenocarpic fruits. This kind of seedless fruits can be generated by making use of hormones, plant growth regulators, about which you have learnt in 11th grade. So you can induce parthenocarpy in certain plants and make them produce seedless fruits. Okay. There are 2 concepts about which you have to learn in this chapter. One is apomixis and the other one is polyembryony. What is apomixis? You know how in sexual reproduction, after fertilization, ovule will become the seed. But apomixis is a type of asexual reproduction which will mimic sexual reproduction, and even though it is asexual reproduction, seed production can be seen. Okay, so there are a few flowering plants such as species of Asteracean grasses. They have evolved a special mechanism to produce seeds even without being fertilized. So basically, seeds which are products of sexual reproduction usually are produced even without sexual reproduction. So it is mimicking sexual reproduction. There are so many ways in which this can be brought about. One example is, in some species, the egg is formed because of meiotic cell division. In some species, egg is diploid itself, meiosis will not take place, and this diploid egg cell, which is formed without reduction division, itself will develop into the embryo without fertilization. So in such cases, seed will form, but fertilization has not taken place. Okay, so that is about apomixis. Talking about polyembryony, what does poly mean? Poly means many. Embryony obviously relates to embryos. So the seeds in which there is more than one embryo, we say it is polyembryonic. So we can see that in some varieties of citrus and mango, what basically happens here is, surrounding the embryo sac, you have the nucellus. Some of the cells in the nucellus, which will surround the embryo sac, they will start undergoing cell division, and they will protrude inside the embryo sac, and they themselves will develop into embryos. In such cases, we can see that within the ovule there is more than one embryo, and such a condition is said to be polyembryony. Okay, so with this we have come to an end of the discussion of this very important chapter called sexual reproduction in flowering plants. I hope all of you have kept up throughout the class and understood every little concept that we've discussed today. All the very best.